This talk was primarily written by Dr. William Seeds. We made it in preparation for the 7th World Parkinson's Congress. We were not selected for a hot topic talk. We thought it would be useful to make the talk available. The topic is the superoxide sentinel hypothesis of mitophagy, a paradigm shift in Parkinson's disease. Rethinking MPTP, mitochondrial quality, and selective mitophagy. The narration is based on a voice actor's voice. Bill Langston worked with the writer John Palfreman to write the book The Case of the Frozen Addicts. There is a new twist in The Case of the Frozen Addicts, the medical mystery. In 1982, several young drug users in Northern California were admitted to hospitals, appearing to be in an advanced state of Parkinson's disease overnight. The toxic culprit. Dr. Langston and others discovered the frozen state was caused by a contaminant called MPTP, a byproduct of a botched attempt to synthesize a designer version of the synthetic heroin meperidine. Discovery of cell death. The researchers found that MPTP specifically targets and destroys neurons in the substantia nigra, the same area of the brain that degenerates in natural Parkinson's disease, providing the first animal model for the condition. What is the new twist in the mystery of MPTP? MPTP itself is not toxic. MPTP is converted to MPP+. MPP+ attacks the healthiest mitochondria. MPTP metabolite. MPP+ is driven to enter and damage the mitochondria with the most negatively charged mitochondrial matrix. These are the healthiest mitochondria. What remains? Weakly charged mitochondria accumulate. The traditional MPTP neurotoxicity model. Hid true discovery. True discovery. Parkinson's is caused by the accumulation of weakly charged mitochondria. Same result, different processes. Accumulating sick mitochondria is not from killing off the healthy mitochondria, but rather a failure in the process that removes sick and weakly charged mitochondria. New mystery. How then are sick mitochondria selected for removal? How does interfering with this process cause Parkinson's? The superoxide sentinel hypothesis of how mitochondria are selected for mitophagy can be summarized in 8 steps. I will go through those steps in this talk. The idea provides a paradigm shift. Old model MPTP toxicity equals Parkinsonian symptoms. New model. Loss of mitochondrial quality control equals disease. Key insight. Dysfunctional mitochondria produce superoxide to signal for their own removal. Consequence. A breakdown in the superoxide-mediated identification process for mitophagy results in a disease state due to the accumulation of damaged mitochondria. Step 1. Superoxide as a sentinel of unfit mitochondria. Molecular oxygen. Dual free radical with unpaired electrons. Superoxide formation. The addition of one electron to molecular oxygen creates superoxide, which has one unpaired electron and a negative charge. Sentinel function. Marks unhealthy mitochondria for removal. Monitored reactions. Reactions that use FAD, FMN, and coenzyme Q. Inside dysfunctional mitochondria can leak an electron to molecular oxygen, thus forming superoxide. Superoxide monitors. This table summarizes the enzymes that interact with the cofactors, FAD and FMN, and the coenzyme Q, to translate from 2 electron transferring hydride ions to sequential 1 electron transfers. The free radical intermediates that enable sequential single electron donations are not normally exposed to oxygen molecules. When there is something wrong with one of the processes being monitored, the free radical intermediate is exposed to oxygen and produces superoxide. This allows all these processes to be monitored by the superoxide sentinel selection process. To follow the narrative, you will need to know more about oxygen, NADH, and NADPi. Why 2 dots on molecular oxygen? This indicates that molecular oxygen has unpaired electrons with the same spin. Physical chemists call this triplet oxygen. Energy constraints prevent triplet oxygen from reacting with proteins, lipids, and DNA. Triplet oxygen can only react with other free radicals such as iron in hemoglobin and FAD, FMN, coenzyme Q, free radical intermediates found in mitochondria, NADH, and NADPH are 2 at a time. Electron donors, they transfer 2 electrons at a time as hydride ions, which have 1 proton and 2 electrons. This allows high-voltage NADH and NADPH to safely diffuse throughout the mitochondrial matrix or cytosol without electron leakage. FAD, FMN, or coenzyme Q Convert mitochondrial electron pair transfers to sequential single electron transfers, forming free radical intermediates that react with triplet oxygen, creating superoxide. Step 2. Messenger deployment. Superoxide dismutase 2, SOD2. Converts 2 superoxide into 1 molecule of oxygen and 1 molecule of hydrogen peroxide. Hydrogen peroxide as a messenger. Exits mitochondria to signal a cellular response to prime mitophagy. Critical limitation. Mitochondria lack catalase, which detoxifies hydrogen peroxide. Strategic design. Lack of catalase allows mitochondrial hydrogen peroxide to escape and signal to the nucleus for a response. Step 3. Activation of a nucleocytoplasmic response. Key responders. DJ1, NF-kappa B, NRF2, calcium 2+, calmodulin nitric oxide synthase isoforms, endothelial inducible neuronal NOs induced or activated. Cofactor requirements. Glutathione, GSH, and tetrahydrobiopterin, BH4B available to NOS. Goal: produce cytoplasmic nitric oxide from arginine. Nitric oxide synthase, NOS, uncoupling. A critical failure point. NOS enzymes work as dimers, 2 copies required. Uncoupled NOS produces superoxide instead of nitric oxide. This superoxide is toxic in the cytoplasm. Requirements to prevent uncoupling include NADPH, GSH, and BH4. The consequence of NOS dimer coupling is the mitophagy signal cannot proceed. Step 4. Nitric oxide production. Nitric oxide characteristics. Nitric oxide is a free radical cytoplasmic gas, but freely permeable into the mitochondrial matrix. Sources include NOS isoforms present in neurons, endothelial cells, and microglia. The key reaction is nitric oxide plus superoxide yields peroxynitrite. Location. Most peroxynitrite is formed inside the mitochondrial matrix where most superoxide is located. Step 5. The mitophagy decision. Threshold determination is based on mitochondrial superoxide concentration. If superoxide in an individual mitochondrion is above threshold, superoxide plus nitric oxide yields peroxynitrite, and the mitophagy cascade is triggered. If superoxide is below threshold, NADPH-fueled antioxidants detoxify free radicals. Quality monitors. Coenzyme Q, FMN, and FAD transfer. Efficiency regulate mitochondrial superoxide generation. Peroxynitrite. The execution signal. Peroxynitrite spontaneously generates carbonate radicals, nitrogen dioxide radicals, and hydroxyl radicals. Peroxynitrite can directly open the mitochondrial permeability transition pore, MPTP, to depolarize the mitochondrion. Antioxidants quench radicals but lose activity in the process. NADPH must restore most antioxidants via electron donation, reduction. Mitochondrial NADPH oxidation to NADPH activates NNT to depolarize the mitochondrion. Step 6. NADPH restoration. Redox potential is determined by the ratios of the concentration of NADP plus to NADH. NNT enzyme transfers a hydride ion from NADH to NADP plus. Trade-off. NNT fueled by proton gradient depolarizes the inner mitochondrial membrane. Dual effect. NADPH reduces oxidized antioxidants, while cytoplasmic NADPH fuels NOS to signal for mitophagy. Step 7. Membrane depolarization. Mitochondrial permeability. Transition pore. MPTP. Opening results from peroxynitrite, which directly triggers mitochondrial pore formation. Proton gradient loss causes the inner mitochondrial membrane potential to collapse. NNT activation contribution. The proton transfer accelerates depolarization. Signal amplification. Depolarization recruits PINK1 and Parkin to the mitochondrion. Step 8. PINK1 Parkin mitophagy. PINK1 accumulates on the outer membrane due to depolarization of the inner mitochondrial membrane. Parkin recruitment results in E3 ubiquitin ligase translocation. The resulting ubiquitination cascade tags mitochondria for identification by autophagosomes. Complete removal. Autophagosomes containing defective mitochondria fuse with the lysosome for degradation. PINK1 and Parkin also activate mitochondrial biogenesis. Key insights into the etiology of Parkinson's disease. PD risk factors may disrupt mitophagy. These risk factors include infection, inflammation, trauma, alpha-synuclein, and toxins. Genetic polymorphisms in genes such as PINK1, Parkin, and DJ1 interrupt the superoxide signaling pathway. The result is that these initiators oxidize cytoplasmic NADPH to inhibit NOS activity or deplete the NAD+ pool, as NAD+ is the precursor of NAD. NADPH deficiency uncouples the dimeric nitric oxide synthase (NOS) isoforms, thereby preventing peroxynitrite formation and selective mitophagy, and reducing mitochondrial biogenesis, which is otherwise initiated by PINK1 and Parkin. Therapeutic implications. The superoxide sentinel hypothesis offers a rationale for treating Parkinson's disease. This includes the following. Remove or mitigate initiators of Parkinson's disease. Stop disrupting mitochondrial quality control with high-carb diets, polyunsaturated seed oils, and unhealthy gut microbiota. Check for individual-specific interruptions in mitochondrial quality control and, if possible, mitigate them. Exercise. Optimize sleep. Practice intermittent fasting. Supplement with any vitamins, minerals, or nutrients that may be missing from your diet. These suggestions are not made to treat or cure Parkinson's disease. They have not been clinically tested in combinations for safety or efficacy. Consult your physician before making changes. References used. You can add your comments to the discussion below.